Hello, everyone. This is Jeremy Yang, data scientist at the University of New Mexico, and with the Illuminating the Druggable Genome Knowledge Management Center, presenting a poster prepared for the February 2021 annual IDG meeting about TIGA, Target Illumination GWAS Analytics. TIGA is an approach, a workflow, and a web app for processing and presenting the evidence from genome-wide association studies, as compiled by the GWAS catalog, assessing confidence for gene trait associations, and for the use case of discovering, illuminating, and prioritizing novel drug targets. TIGA has been a collaborative effort aimed at providing a pipeline for incorporating GWAS evidence into the IDG Target Central Resource Database, TCRD, and the IDG Knowledge Portal, FAROS. In addition, the diseases resource from the Lars Juul Jensen lab is incorporating TIGA for its experimental channel. In this presentation, I'll summarize the TIGA approach and then offer some options for learning more, including a manuscript preprint currently under review and revision. A separate video will demo the web app. Regarding GWAS in general, there is a strong consensus that GWAS offer great value for revealing the genomic basis of traits, including diseases, but also that GWAS present numerous difficulties in areas including study design, instrumentation technologies, and statistics of analysis. TIGA addresses the IDG use case, simplifying our task by narrowing the focus to protein coding genes and drug target prioritization. TIGA depends exclusively for its GWAS data upon the NHGRI EBI GWAS catalog, a great resource and project which curates and harmonizes GWAS metadata and summary data with frequent updates. These plots indicate the dramatic growth and progress of GWAS, including evolution of technologies, and particularly from Illumina. Varying sample sizes affect statistical power and association confidence. The table lists traits which GWAS indicates are highly polygenic, perhaps too polygenic to be particularly useful, but informing future study design and interpretation. Drug target illumination requires focus on conditions amenable to drug therapies. From biologists, we learn how the very definition of a gene has evolved. TIGA focuses on protein coding genes in accordance with current understandings of target druggability. But we understand the foundational science is evolving. Aggregation is an important word relating to aggregate statistics, but it can be quite challenging to aggregate instances of entities such as SNPs, genes, proteins, and traits rigorously and appropriately, requiring identifier resolution and plenty of data wrangling. Aggregation also depends on semantics and ontology, as in this example, where traits may be grouped as related or by a subsuming trait. In order to assess evidence for gene trait associations, we have developed a number of aggregate statistics to count or measure instances of specific evidence. This reflects an axiom of empirical science that additional instances of independent confirmatory evidence increase confidence. For example, N study is the count of distinct studies supporting a given gene trait association. And SNP is the count of unique SNPs for all studies supporting a given gene trait association. All such measures are designed to be non-controversial and scientifically intuitive. Of special importance is RCRAS, which incorporates the bibliometric relative citation ratio. The National Library of Medicine iCite RCR was published in 2016 and measures the evolving impact of an individual publication relative to its scientific area, in contrast to the less specific and more static journal impact factor. By aggregating RCRs into the RCR aggregate score, RCRAS, we seek a corresponding measure of scientific consensus for gene trait associations. Since TIGA is designed to reflect experimental findings, it might be questioned whether a bibliometric component is justified. However, scientific findings, including GWAS, are filtered and mediated by peer-reviewed publications, explicitly so in the GWAS catalog. So in this context, we claim that RCRAS is simply a way to refine and improve on this existing necessity. From the aggregate statistics, we developed a scoring algorithm from a selected subset of variables. To avoid ad hoc weighting, mean rank was employed and then normalized to a mean rank score with range 0 to 100. Variables were selected by benchmark against the gold standard gene disease association set based on the genetics home reference and also used by the Jensen Lab Diseases resource. This is a screenshot of the TIGA web app available publicly, which supports trait queries, gene queries, and gene trait association details and provenance. Full dataset download is also available, and there should be a separate video demo for the TIGA web app. To learn more, you can read our manuscript in review and revision at bioRxiv and/or check out the web app online help or public GitHub repository. TIGA is built upon the strengths of the NHGRI EBI GWAS catalog and designed to serve the IDG community as a standalone web app and workflow available via GitHub repository, but mostly via the IDG portal Pharos and its backend database, TCRD. TIGA is simultaneously designed to provide experimental channel evidence for the Jensen Lab Diseases resource. Through these channels, TIGA is intended to contribute to drug discovery research by helping to shed light on the understudied druggable genome.